Okay, so in this lecture we're going to talk about translation, and we have a picture here of the structure of the ribosome. We have the large subunit of the ribosome shown over here on the left, and the small subunit. So you've seen this slide before, and we've talked about different parts of it. We've talked about replicating DNA using the enzyme DNA polymerase to copy the genetic material, DNA. We've talked about how RNA polymerase can take the DNA, copy it into RNAs through the process of transcription. Now, we have different kinds of RNAs that are shown here. These all now are going to funnel into the ribosome, which will take these RNAs and use them to translate the nucleotide sequence in the messenger RNA into an amino acid sequence of proteins. And so that's what we're going to talk about today. Okay, so we're going to start with looking at an experiment that happened some time ago in the early 40s, work done by George Beadle and Edward Tatum. They were interested in the relationship between genes, enzymes, and traits. And they were asking a very specific question. Does one gene encode one enzyme, or does one gene encode many enzymes? So they used a model organism, common bread mold, called Neurospora crassa. This was useful because they could look at simple nutritional requirements. So they had several strains, upwards of 2,000 strains. Now these strains had been, these are mutant strains that have been produced by irradiating Neurospora to generate mutations. They analyzed the different enzyme pathways for the synthesis of vitamins. So here's a sort of a schematic of their experiment. So what we have is here we have minimal media plate, and wild-type Neurospora grows just fine on that. Now they had worked all these mutations down into 4 groups, presumably by complementation analysis, which we talked about earlier in the semester. And none of these mutants could grow on just minimal media. But they knew there were certain compounds in the pathway for synthesizing methionine. And they would add each of these compounds to different plates and see which one of these strains or which strains would grow. So in this first case, they added O-acetylhomoserine. When they saw that, when they did that, they saw that one mutant grew on there. When they added the other compounds, they saw that that same mutant also grew. So this particular mutant 1, could grow when as long as one of these compounds was added, it would be able to grow. Now that was not true for all of the strains, because as we can see here, adding O-acetylhomoserine did not allow growth for mutant number 2. However, adding cystathionine did, and then adding homocysteine and methionine allowed number 2 to grow. Mutant number 3 wouldn't grow with cystathionine; it needed homocysteine and also or methionine. And then the last one wouldn't grow at all unless you just gave it methionine. So they looked at these results, and based on them, they felt they could determine the order of the enzymes in the pathway. So they already determined how many enzymes were in the pathway. Now they want to see what's the order. And so this is their model. And the model is that there are 4 enzymes. Each has a certain reactant that it uses to produce a product. And as you go down this pathway, the second enzyme will use the product of the first one as a reactant. The third would use the product from the second one as a reactant, and so forth. Okay, so what's going on here? So they noticed that by adding O-acetylhomoserine that mutant number 1 could grow. And it could grow whether you added any of the other products in the pathway. So what's going on there? So here, if you reason this out, if enzyme 1 is defective, that means it can't take the ultimate precursor, the very starting material, homoserine, and convert it to O-acetylhomoserine. So it can't do that. However, if you provide this compound to the media, then enzyme 2 can take this and convert it to the next product. Then enzyme 3 can do its job, and so can enzyme 4. But this should also be true for any of these other compounds further down in the pathway. So by adding cystathionine, enzyme 2 doesn't do anything, but enzyme 3 can now pick up at this state and generate its product, homocysteine, and then enzyme 4 can convert it to methionine. All right, so that's what's happening here. So if you look at the sort of pattern that comes out of this, every time that you have an enzyme that is defective in one of these reactions, if you give it a compound that's downstream from its position in this pathway, then the remaining enzymes can finish off this pathway. So in doing this, we could determine that the first enzyme needed to make this O-acetylhomoserine, second one needed to make cystathionine, the third one had to make homocysteine, and the last one took this and converted it to methionine. So they determined that every mutant strain was blocked at a particular step in the synthesis pathway, showing that each gene encoded one enzyme. So we've learned quite a bit since then, and we know that there are exceptions to this one gene, one enzyme hypothesis. The first is that enzymes are only one category of proteins. Some proteins are actually composed of more than one polypeptide. So you need more than one, 2 or 3 or 4 or more polypeptides to make the functional protein. Many genes have been identified that don't even encode proteins or polypeptides. There have been several non-coding RNAs that have been identified, and we're going to talk about a couple involved in translation, which is transfer RNA and ribosomal RNA. And we've also talked previously that one gene can encode multiple polypeptides through alternative splicing. Okay, so let's start talking about translation and how proteins get made. So translation is the biological polymerization of amino acids into polypeptide chains. The process requires messenger RNA, and we've talked about transcription and how RNAs are made, amino acids, a genetic code, transfer RNA or tRNAs, and ribosomes, which also contain another form of RNA called ribosome. So DNA and RNA are polymers composed of nucleotides. Proteins or polypeptides are polymers of amino acids. And so here is the structure of amino acids, general structure. We have an amino group, shown here in blue, the carboxyl group in red. These are both attached to a central carbon called the alpha carbon. Also attached to the alpha carbon is a hydrogen and a variable side chain designated as R. If we look at the 20 common amino acids, we can see this common portion shown here at the top of each of these little pictures, and we can see the side chain shown in yellow for each of them. As you can see, there's quite a bit of variability. Now, these amino acids can be classified into various ways. So, some that you should know. You should know that about the basic amino acids, arginine, lysine, and histidine. You should know about the acidic amino acids. They're easy to remember because they have acid in their name, glutamic acid and aspartic acid. And I'm going to use these, when talking about genetic code, I'm going to use these in a later slide as an example, so keep those guys in mind. You don't have to know all the polar amino acids because there's several of those, but there are polar amino acids. Then there are hydrophobic amino acids, and again, you don't have to remember all of those. But there are multiple ways to classify amino acids, and in addition to just the way I've described them here, there are also aromatic amino acids, and those you should know. Which ones are the aromatic amino acids? Tyrosine, phenylalanine, and tryptophan. And they have these sort of ring structures. There are other amino acids that are incorporated enzymatically at stop codons, selenocysteine and pyrrolysine. Okay, so what happens when you take a couple of these amino acids and you stick them together? these amino acids form peptides. So here we have one amino acid 1 designated by the side chain here, R1, and amino acid 2 designated by the side chain, R2. Now, they're going to come together here, and you can see this portion highlighted in blue right here is basically a water molecule. So that water molecule gets removed, and these two amino acids get stuck together. They form this peptide bond, and we have this peptide group here. Now, since there are two amino acids stuck together, this is called a dipeptide. And this can go up to, you imagine, polypeptides are sort of chains of several amino acids. And they can look like this. So you just have multiple peptide bonds formed. But there's something else that's kind of distinct about this. So here we have 4 amino acids, 1, 2, 3, 4. On one end, the amino group from this first amino acid is intact. On the other end, the carboxyl group is intact. So these would be the amino terminus, or N terminus, and the carboxyl terminus, or C terminus. And so each polypeptide chain is directional, having an N terminus and a C terminus. You can also look at proteins different levels of structure. Now, what I've just shown you is the linking of these amino acids to form the polypeptide. That's the primary structure, the order of the amino acids and how they're linked together. Now, this order of amino acids dictates the next level of structure known as the secondary structure. So amino acids can fold around themselves or upon themselves and forming hydrogen bonds with each other to form some distinct structures. One is an alpha helix, so that's shown here, this sort of ribbon, and then beta strands, which are shown here individually, which come together to form beta sheets. Now, these secondary structures, and I should mention before I move on, there are also other, these are the two predominant secondary structures. There are also loops and also unstructured regions in proteins. Now, how all those come together and form from one polypeptide and form a three-dimensional structure, that is known as the tertiary structure. And so here we have an alpha helix, we have some beta strands forming a beta sheet and some loops and possibly some unstructured regions, and they form a very distinct structure. Now the last level of structure, not all proteins have this. The quaternary structure is only applicable to proteins where more than one polypeptide has to come together to make the functional protein. Now the polypeptide can be identical polypeptides, so two of the same peptide or more coming together to form the active protein, or it can be made up of many different polypeptides. And so this is just showing here that we have in this case, two identical peptides coming together to form the final active protein structure. And when you look at proteins, right, again, there are these long chains of amino acids. They can be hundreds or thousands of amino acids long, and they have quite a variety of three-dimensional structures that can form. This bovine rhodopsin here is mostly alpha helices, so they're sort of shown as these sort of curly Q French fry looking things here. Right, these are your alpha helices, and there are some loops joining them together. If we look at this nucleotide transporter, we see that there's lots of beta strands, and they're often depicted as an arrow. It's easy to see this one right here, indicating their direction. And so the beta strands come together to form a beta sheet. These actually form a kind of tube known as a beta barrel, which will oftentimes these put holes in membranes and allow transport of different kinds of molecules. Okay, so the genetic code is another hugely important piece of translation, and we're going to talk about this in detail. So the genetic code is written in a linear form using ribonucleotide bases that are in the messenger RNA. So the bases, the GAs, U's, and C's are the letters. Now, these letters come together to make up words, and what we now know is that the words are composed of 3 nucleotides, right? And those 3 nucleotides will code for amino acid. So if you look at the messenger RNA, so here we have the 5' end of the messenger RNA and the 3' end of the messenger RNA. These correspond to the N-terminal region of the protein will be closer to the 5' end, and the C-terminal portion will be closer to the 3' end. So the nucleotide sequence and amino acid sequence are considered collinear. Right? There's a relationship between the sequence of the nucleotides in the messenger RNA and the sequence of the amino acids in the protein. Okay, so how does this code work? Okay, so determining what the genetic code consists of was a very big issue, and it took a lot of work to decipher this. And we'll talk about a couple of the experiments. This is not the only experiments that were done to figure out this genetic code, and it took some amount of time. Okay, so the question is, how do nucleotides specify 20 amino acids, right? There are only 4 different nucleotides in an RNA, A, G, C, or U, right? That's it. So obviously there has to be some sort of code. Now, it can't simply be a 1-letter code. Like A cannot, there are only 4 combinations, right? You can either have A, G, C, or U. That is less than 20 amino acids that need to be encoded. If the code is 2 letters, well, at each position you could have 4 letters, any of the 4 letters, and at the first position and the second position you can have any of the 4 letters. They are independent of each other, so you just simply multiply at each position. You have 4 times. At the first position, 4 times. Second position, you get 16 total combinations. Again, that's less than 20. That's clearly not going to do it. However, when you reach a 3-letter code, each position has a chance to have one of the nucleotides. That comes out to 64 possibilities, which is much greater than the 20 that we would need to code for the 20 amino acids. So this is fortunate, right? So based on this, researchers hypothesized that there's a 3-letter code, which is called a codon, with 64 possibilities for all 20 amino acids. So that means more than one codon can code for the same amino acid. And there's this term for this, the genetic code is degenerate. It's not a bad genetic code, it's just there are multiple ways to code for the okay, so this led to Nobel Prize in 1968 for deciphering this code. Robert Holley from Koreana and from Nuremberg. So they had to use some tools to do this. It definitely was not a straightforward kind of problem. And so they used some more biochemical tools to do this. And they asked the question, what is the nucleotide code for each amino acid? And the tool that they used were these. They had the ability to synthesize RNA. Right? So they did this enzymatically outside of a cell. So this is biochemistry. Then the enzyme polynucleotide phosphorylase. That just takes ribonucleoside diphosphates, so there's only 2 phosphates, and you mix those in with an existing chain, or you just start putting in a whole bunch of these ribonucleoside diphosphates, and eventually you're going to get an extension of this chain. You're going to get more of these nucleotides. Right? So they could synthesize a synthetic RNA. Now, this didn't have all the start signals, and actually the sequence of these RNAs is pretty much random. They didn't have any control over which nucleotides got put in. There was no template to designate which nucleotide is going to go in which position. So they were clever about this, and we're going to talk about what they did to sort of use this. Now, the other thing that they had was what's called a cell-free or in vitro protein synthesizing system. And I'll talk about that on the next slide in some detail. Okay, so here's the sort of the idea behind a cell-free protein synthesis. And so people have, before these guys used it, people have been making these, so they weren't completely new at this time. But what they would do is they would grow up cells, for example, E. coli, in a flask. And so in there, this is just showing you, here's a cell, and all these goodies, ribosomes and everything else, RNAs, transfer RNAs, amino acids, everything's inside this cell. And so you got a whole bunch of cells growing up in here. They would break open the cells, which would release the contents. You get all the goodies out, and you're going to use them. Now, the problem is there's more in there than they actually want to use. So they use a technique of centrifugation, usually in some sort of density gradient, and I think it was usually sucrose. And they, in centrifuging the contents of the cell through one of these gradients, they could separate different parts of the cell. So they wanted to get rid of messenger RNAs because they wanted to give their own messenger RNA, which is the one that they were synthesizing, shown in the previous slide. They want to put in the amino acids, and that's going to be very important. They want to get rid of DNA and RNA polymerases, so no new RNAs could be synthesized, but they wanted to keep some things. They wanted to keep the ability to synthesize protein, so they had transfer RNAs and ribosomes. Right? And so this is what was in the cell-free protein synthesis system. So they break open the cells, they spin them in a centrifuge, take certain portions of that, the liquid that was spun down, and then they could separate out these parts, and they put all those into a tube. So then to start their experiment, they would add the other components. So they didn't have amino acids, so they would add all the amino acids, but they would have one amino acid that was radioactive. And we'll talk about this over the next few slides, why this is really significant. They would also add in their RNA. And so they were clever for starting this. Instead of having all 4 nucleotides in a message, which will give you a multitude of combinations of codons, they said, why don't we use just one? And they used just uracil. And so they had a synthetic messenger RNA made up of just uracil called poly-U. So the rationale here is that whatever the code, whatever amino acid gets incorporated has to be coded by 3 U's, because that's all that's there. To me, that initial step is really just a clever step. So they did this with all the amino acids, and in each case they would swap out with a different radioactive, one amino acid being radioactively labeled. And again, in the next couple slides, I'll tell you about how they're able to detect that. But when they put in this RNA with radioactive phenylalanine, they were able to detect protein synthesis, and it was made up of the proteins were made up of all phenylalanine. And so they've learned what one of the codes is. They learned that UUU codes for phenylalanine. All right, so these RNAs that they used are called homopolymers because they consist of only one nucleotide. Right, so there are 4 different ways of doing that. You can have all U, all G, all A, and all C. And you mix those with the in vitro translation system. And as I mentioned, they have 19 amino acids that are not radioactively labeled, and one of them is radioactively labeled. And they used carbon-14 or C14 for this. And they could detect the incorporation of the radioactivity. So that's what they're looking at. So they tried for each of these homopolymers, and so they did for U, A, and C. They tried G, but G gave them some problems, so they had trouble with that. And they asked which amino acids get incorporated, which are shown right here. And when they looked at the results, they found that when they had radioactive phenylalanine, that would get incorporated in poly-U, but they also checked to make sure it didn't just get incorporated in any of the other homopolymers, messenger RNAs. And so they didn't put in any RNA, and they got a certain level of radioactivity, very low. They got a very high number when they put in the poly-U, but then they essentially got background levels when they put in A or C. So they knew that it was very specific for the poly-U for phenylalanine. Right? So they cracked these 3 codons. What about the rest? And that's where things start to get a bit more complicated. Okay, so we just talked about how you would do an experiment where you had an RNA that had all the same nucleotide. So what happens if you want to synthesize an RNA from a mixture of bases? So we'll get how that was done in a predictable fashion. But I'm going to talk about the details from of how the experiment was performed. And keep in mind that this is how the experiment was performed when you used a single nucleotide to make up the RNAs. So at an experimental level, we have the cell-free translation system in 20 different tubes. Now, any RNA that you put in there, you need to synthesize and mix into these tubes. Now, if it's one nucleotide, you can predict what the codon is going to be. And it's only going to be composed of one nucleotide. But you have no way to know what the actual sequence of the messenger RNA is going to be if you have a mixture. And so this is what they did. But keep in mind, this sort of RNA, this can either be, you can swap out which RNA you want to put in here. It can either be a mixture of nucleotides, or it can just have a single nucleotide. So this applies to the experiment that we just spent some time talking about, and with the one we're going to talk about. So you just take one radio-labeled nucleotide and the other 19, radio-labeled amino acid. We are talking about protein synthesis here. In this case, it's glycine. And the other 19 amino acids are not labeled. And you mix those in with the cell-free system and the RNA that you put in, and you incubate it at a certain temperature. 37 degrees is often used. And the RNA, again, this is just a random portion of the RNA with a particular sequence. And if you get glycine incorporated, it's radioactive. It will get incorporated into polypeptide chain, and that polypeptide will become radioactive. Now, 2 things are going on once that happens. Right, so polypeptide will be radioactive, and you need to capture that polypeptide. And the way that's done is using a chemical called trichloroacetic acid, or TCA. And when you add that to this solution, any polypeptides that it forms, so longer chains of amino acids, will do what's called precipitate. They will come out of solution, and they will become and no longer soluble. You take that and you dump it onto a filter paper. So here's a filter paper on top of a vacuum source. And what that vacuum does is it pulls all the liquid through. Now, if you have polypeptides, they will get stuck on this filter paper, on the top of this filter paper. But the amino acids, whether they're radioactive or not, will get pulled through. So all you're going to see here when you look at this is radioactive proteins. Now, how do you look at it? Well, you take this filter paper after you've washed it a couple times to make sure all of the unincorporated amino acids get washed through, and you truly only have the polypeptides on the top of the filter paper, and you put it in a vial with reagent called scintillation fluid, and you put it into a piece of equipment called a scintillation counter, and it can measure the decay of the radioactivity, and you can basically see how much radioactivity is in or is on this filter paper. And keep in mind that the only thing that's going to be on here are polypeptides that have been retained by the filter. Now, if you used an amino acid that did not get incorporated, you would still get polypeptides retained on top of this, but they wouldn't be radioactive. So you could directly measure how much radioactivity was incorporated into proteins. And I said that. Now, this is, keep in mind, this is done really well in your book. Okay, so mixing the nucleotides. I mentioned that you can't just sort of haphazardly mix them. You have to do it in some predictable manner. And what they did is they took 70% of G's and 30% of U's. And so in doing that, they could predict the frequency of each triplet that would occur in an RNA. Keep in mind that the incorporation of each nucleotide is independent of the previously incorporated nucleotides. So what are the possibilities? So you have 0.7 or 70% chance of a G getting incorporated at a position. So that's shown here. Now, the chance of a G getting incorporated at the next position is also 0.7. And if you, in order to get at the third position, that will also be 0.7. These are all independent events, so you multiply them and you get 0.34 or 34%. So 34% of the triplets in mRNA or RNA made where you have 70% are G and 30% are U, 34% of those triplets will be GGG. You can figure this out for GGU. Again, you would have 70% chance of getting G at the first position, 70% chance of getting it at the second position, but only a 30% chance of getting it at the third position. So that's going to come out to be 15%. And then you can see, same reasoning. You have, 1G, 70%, 2 U's, they're each going to be 30% each. You come out to 6%. Now, the one that we know about, the chance of getting UUU, which we already know encodes phenylalanine, there's going to be a 3% chance of that happening. So you figure out all these different permutations, and they will add up to 100%. Okay, so they do the experiment, and then they ask what's the percent of each polypeptide that they get that it's after incorporating glycine, valine, tryptophan, etc. Different amino acids. And so they get these different numbers. Now, these numbers don't necessarily match up to exactly what we predicted. So what's going on here? Well, luckily, phenylalanine does. We predicted that there would be 3% of the codons would be phenylalanine, and that's what was actually observed. But what about these other ones? So this requires multiple experiments, what I'm about to tell you. And can't all be derived directly from just this one experiment. But this glycine, we know that GGG goes for 34%, but then some other triplet that is predicted to occur 15% of the time is going to account for the remainder of this 49%. And we now know that that other triplet is GGU. For cysteine and leucine, they are each specified by a codon that has 1 guanine and 2 uracils. So we get 6% for either of those, but we can't actually at this point tell which triplet codes for either of them, right? And so just to reiterate that this is a single experiment. This was done multiple times. If you start thinking about the amount of work that went into this, it's astronomical, especially by today's standards. But you had to combine work from multiple experiments to sort of figure this out. And now, one of the next experiments they'll talk about help to really decipher which triplet exactly corresponds to which incorporated amino acid. And that assay is the RNA triplet binding assay. So what's done here? So here we have a ribosome now. So ribosome is in there. And this is purified components. We have particular transfer RNAs with radioactive amino acids on them, filter paper again, and then we have, right, so we have one transfer RNA that's radio-labeled. You have to do this separately for each amino acid. And then we have a 3 nucleotide RNA. So that's why they're called triplet RNA. So you have to have each one of these separately. So you start seeing that the combinations are pretty large that you can get here. And the idea is that if this short RNA can actually interact with the ribosome, the transfer RNA can interact with that RNA. And if it does, it will then be retained by this whole complex of the ribosome and the transfer RNA and the small, tiny messenger RNA, triplet RNA that's in here. And that will be retained on the filter all these other things will get washed through, all the unincorporated transfer RNA with its radioactivity, so you only see if they are bound. And so this would use the same sort of scintillation apparatus to scintillation counter to measure this. Right? So what we end up getting is something that looks like this. We have amino acid with the labeled proline sitting at a particular triplet. And this tells us exactly which tRNA is bound to exactly which codon. Okay, so they did a lot of work. Right? This was a massive undertaking, but it was important, and we learned a lot of things about the genetic code. So what we learned is that through multiple experiments, we verified that it was, or they verified, I didn't do anything, they verified that it is a triplet code. Each 3 nucleotide codon in mRNA specifies 1 amino acid. The code is comma-free. That means the mRNA is read continuously, 3 bases at a time without skipping bases. Now, there have been exceptions to this that have come up, and those exceptions are translational frame shifting. It's not overlapping. Each nucleotide is part of only 1 codon and is read only once. We mentioned that it's degenerate. 18 of the 20 amino acids are coded by more than 1 codon. Methionine and tryptophan are the only exceptions. There are start and stop signals. ATG is generally the start codon for a polypeptide. It also codes for the amino acid methionine. Then there are 3 stop codons that can be used to terminate translation. There is a phenomenon called wobble, which we'll talk about. This occurs in the transfer RNA anticodon, the part that reads the codon. In the 3rd base of the codon is less constrained and pairs less specifically. We'll talk about that in more detail. And that this code is almost universal. Most codons have the same meaning in all different organisms, but there are some exceptions. Okay, so all that work went into figuring out the genetic code, and we learned all those different things. So here is what the genetic code looks like. So it can be summed up in this genetic code table. So how do we use this table? So each amino acid is sort of designated here: phenylalanine, leucine, isoleucine, methionine, valine. Okay, so they're all sort of indicated. Your stops are here, and your starts are here. So this is the codon read from the 5' end to the 3' end. So remember, 5' is always on the left. And if you look at the sort of outer part of this table, it tells you what you can what nucleotides are in which position of the table. So okay, so this is the first position. The 5' end is indicated on the left here. So that means if you look at all the codons that are sort of in this row here, they all start with U. So if you want to find something that starts with U, that's where you would look. It's easy to find. Now for the across the top tells you what's in the 2nd position, and you can see here that there are U's all the way down in the 2nd position. Here there's C's all the way down in the 2nd position. Now on the right hand side, this tells you for each row what's going to be in the 3rd position. So here U's are in the 3rd position, then a C, and then this gets repeated for each of these different sort of 1st position rows. So that's how you look. If you wanted to know which amino acid was encoded for by GCU, for example, you would look here, 1st position, say where's G. All right, so G's here. C in the 2nd position is going to be somewhere in here, and we see GCU is going to be alanine. All right, so that's how you use this codon table. So what are some things we can see in this table? So here is our start codon, and it's also the methionine codon. Our stop codons are located up here. All right, so they're all sort of clustered together. Many amino acids are encoded by multiple codons because the code is degenerate. This is something I've been saying from the beginning. And so here's an example. So here we have alanine, as just mentioned, coded by GC, and something else. So the first 2 positions are fixed for alanine. Anything that has a G or C, G in the 1st position, C in the 2nd position is going to encode alanine. But the 3rd position can change. Now this protects against radical mutations. So if there happens to be a mutation happening at this third position from a U to a C, well, it's not going to make any difference in the protein. It's still going to code for alanine. So let's look at another place. So these are sometimes termed as boxes. So we're going to look at this box. Now this box is split. So these are the two acidic amino acids, aspartic acid and glutamic acid. And here are the codons for each. Now these are similar amino acids. They have similar chemical properties. They both have GA in the first position. Aspartic has pyrimidines, right? A U or a C in its third position. Glutamic acid has purines in its third position, either an A or a G. This again protects against radical mutations, because even if there's a mutation, so it's a mutation from U to C, it's still going to be aspartic acid. But if there's a mutation from U to A, well, it's still going to be an acidic amino acid. It's still going to have very similar properties. And if you look throughout this code, you'll see lots of other examples of this. Right, so there are many changes that result in chemically similar amino acids. All right, so there's order to this code, and there's a couple things that I'd like to just mention. So the first position often changes the amino acid. So if you a change in the first position often changes the amino acid. A change in the second position always changes the amino acid. And a change in the third position. All right, so I mentioned that there are certain changes. So these are mutations. And we're talking, the changes that we've been talking about are point mutations. So single base pair changes that occur in the DNA that change the sequence of the messenger RNA that can have an effect on the amino acid that is coded. Now, there are silent mutations where the change happens, but it doesn't change the amino acid that's coded for. So we had the example of GCA getting changed to GCU, still codes for the same amino acid, alanine. There are missense mutations, which a change in the codon actually results in a different amino acid that will get incorporated into the protein. And then there are nonsense mutations. Now these are mutations that result in a change from a particular amino acid to a stop codon. So it terminates protein synthesis prematurely. The other kind of class of mutations that we're going to consider here are frame shift mutations. These are when you get an insertion or a deletion of nucleotides. The previous point mutations were the same number of nucleotides were in the coding sequence for the protein. Just one of them gets changed. Here we either add a nucleotide that wasn't there originally in between two other nucleotides, or you can have deletion of a nucleotide, and you can have addition and deletion of multiple nucleotides. Now, this is going to have a profound effect as long as it's not a multiple of 3. Multiple of 3 oftentimes is tolerated very well because essentially you're adding in a codon's worth of a material. But other kinds of insertions and deletions will have this sort of frame shift effect. So what's going on? Let's take a look at these. Let's take a look at these types of mutations specifically. So here I have a messenger RNA sequence. It has a start codon, has a stop codon, and it codes for these amino acids written on top. So what happens when this position gets changed, right, to C? If we look back at our code, we'll see that changing this U to this C still codes for histidine. So this is the example of a silent mutation. The amino acid is not changed. This is also, again, at the third position of the codon. Here, this U is getting mutated to a G. Now, all of a sudden, this leucine this codon that codes for leucine now codes for valine, so a different amino acid gets incorporated into this protein. Nice thing about this mutation is that leucine and valine are both hydrophobic and very similar kinds of amino acids. And the codon table, when you look at it, you'll see that there are, again, lots of places where even when the amino acid is changed, it changes to a different amino acid that has very similar properties. Now this next example, this C in the serine codon is changed to an A. This is one of our stop codons. So now the only protein that will be made will be met, gly, his, and lu, and the rest of these will not be made. The rest of these codons will not be read and added on. So, well, this is another stop codon. So this arginine and this serine will not, or so this arginine and this serine will not be incorporated into this nonsense mutation. This nonsense mutation causes premature stoppage of translation after this leucine codon. Okay, so the last example is a frame shift example, and we have some more slides to sort of go into this in more detail. So here, there is an extra U has been added, right? So it's inserted here. So there's a bunch of U's, and all of a sudden we have one more U. So what happens there? Well, it changes the way that this sequence is read. The codons are now all sort of shifted over. The same, most of the sequence is still there, but now everything's off by one. This is known as the reading frame, and the reading frame has been changed. So the reading frame was starting with AUG, GGA, CAU. Then up here we would go to UAA is the next one, but now all of a sudden we have UUU. But that UA is still there. You can see it right here. But now the ribosome reads every 3 nucleotides. It's going to read these 3, then these 3, then these 3, and so on. And so you can see that now that this amino acid is changed after this insertion, but so are a bunch of the other amino acids. Okay, this can be demonstrated very, very nicely with this sort of sentence here of 3-letter words. The big boy saw the new cat eat the hot dog. Right? So when we look at what's happening with frame shifts, we can use this as an example. If a nucleotide is added, right? So in this case, another T, we see that up until that nucleotide is added, the sentence reads the same. Now all of a sudden, if we're reading 3-letter words, now we have TTH, which is no word, ENE, which is no word. So the reading frame is disrupted. Okay. Keep in mind that all this information is still in there, but now it's out of what's called out of register. It's out of frame. Same thing happens if you delete one. We get out of frame. However, if one gets inserted and then another one later on gets deleted, the net sum of this is 0. And you can see that the reading frame is intact up until the first mutation. Then it gets a little wonky. There's a different sequence here. And then there's another deletion that compensates for this addition, a subtraction of 1 nucleotide, and the reading frame gets restored. So the protein, if this was a protein, this would be fine for this portion. There'd be something wrong in the middle, and then you would have the correct sequence afterwards. And depending on the length of this and the nature of this could be really bad, or sometimes it has very little effect. But this is called a revertant. The original mutation it gets sort of canceled by a secondary mutation. It reverts the reading frame back to the original reading frame. This doesn't work if we add one here and then we add another one later. We still have a disrupted reading frame. This is two nucleotides, or in this case letters, have been added. So we still can't read this. However, you can also get another revertant where you have your first mutation is an addition. Then you get a second addition. It's still wrong. The third addition, well, now all of a sudden you've added 3 nucleotides. This is the equivalent of a codon. Now, even though they're not all at exactly the same location, or within one codon space, or adjacent to each other, they still end up restoring the reading frame, and you can read the portion after this will now read as normal. Okay, so now what happens when we actually look at nucleotide sequences and amino acid sequences? So there are multiple reading frames that occur. So in any DNA sequence, you can start reading at a particular nucleotide, or the ribosome can start reading a particular nucleotide, and it will read in groups of 3, right? When you translate this, you transcribe this into RNA, and you will get, in this case, CUC, AGC, GUU, ACC, which will encode for leucine, serine, valine, and threonine. Now, if you just look at this sequence, what happens if the starting point is actually this U? So we're in a different reading frame. So we have UCA, this is going to be serine, then alanine, leucine, and proline. So we're going to get a different amino acid sequence. And there's a third reading frame, which is starting on this last position in this, what was originally this first codon. Now we're going to call this the beginning of a codon. This will be the first position down here, and we get glutamine, arginine, tyrosine, and histidine. We get yet another sequence of amino acids would be incorporated. So this reading frame is designated by the ribosome and the start codon and that interaction, and we'll talk about that very soon, because the ribosome can bind to an RNA, and so translating at any given position, you might end up with multiple different proteins being made, but there are very distinct start signals for this. Now, these are 3 reading frames that are designated on the forward or top strand. However, you can have 3 more that are on the reverse strand. These will be read in the reverse complement orientation. I'm only showing, at the end of this, there'll be only 4 of them. I'll give you one example here. So if the DNA is read and it transcribes the opposite strand, and I've just written that on top here, now it's going 5' on the right-hand side, and reading the codons going in the reverse complement orientation, we will get different amino acids incorporated, and it could be a distinct protein. So in any piece of DNA, there are 6 possible open reading frames, what they're called. So you can read in 6 different registers, 3 on one strand, and then 3 on the other strand. Okay, so another thing that we sort of mentioned before is that the genetic code is nearly universal. So here's our genetic code table, and there are some exceptions in some organisms that to what these codes mean. So one example, so AUA here, is isoleucine is the sort of the universal meaning in most organisms, but in some yeast and mammalian mitochondria, this codes for methionine. Now this, if you look at it, makes kind of sense because it's right next to methionine. It has the same first 2 letters in its codon. The first 2 positions are the same, and it only differs by purines. So that makes sense. That could code for methionine. And then another example that I want to sort of show is here we have the stop codon that is tryptophan in mammalian mitochondria. And again, for the same reasoning, it makes sense that this would code for tryptophan. It's right next to tryptophan and only differs in its 3rd position. And it's either an exchange of a purine. Okay, so we just talked about the genetic code. So how is the genetic code actually read? So the genetic code is decoded by transfer RNAs. So transfer RNAs, or tRNAs, are 75 to 90 nucleotides long. They're an adapter between the messenger RNA triplet code and amino acids. Now this whole concept was first proposed by Francis Crick. The tRNAs recognize the codon in the mRNA through base pairing. All right, so this is a representation of a transfer RNA, this little ribbon with this ball. Transfer RNAs are named based on the amino acid that they have on them. So the ball is the amino acid. In this case, this is phenylalanine attached to the 3' end of the transfer RNA. This would be a phenylalanine transfer RNA. It has some positions in it that we'll talk about in more detail, but this position is really important, where on the 3' end where the amino acid is attached, and the anticodon, the part that's going to read the messenger RNA. So there's another example. Here's proline, transfer RNA. It has proline attached on the 3' end. Okay, so this anticodon is anti-parallel to the codon. So here's the messenger RNA. This is, we're going from 5' to 3'. So we have a codon for phenylalanine here, a codon for proline here. This sequence is going to be read 5' is over here on the right, the 3' on the left. Right? So this is the 5' position of each of the anticodons in the transfer RNA. That's going to match up, or base pair. Okay, so here is the structure of the transfer RNA, and there are some points that we should talk about. First thing we should look at, the overall structure is this cloverleaf structure. Now this is just a secondary structure or two-dimensional representation. So as with proteins, RNAs can have secondary structures and three-dimensional tertiary structures. And this is just the secondary structure showing regions of base pairing for stems and then regions that are not in base pairing as loops and bulges in various places. Okay, right. These are our base paired regions, our loops where there's no base pairing. Now this part I mentioned is important. This is the 3' end. So we start here on the 5' end. There it goes around. Here's our anticodon coming around here to the 3' end. This is important. This is the place. These 3 nucleotides are in every transfer RNA, and this is the place on the transfer RNA where the amino acid is attached. So here's a covalent bond between the transfer RNA and the amino acid. So that's a very important part of this transfer RNA. Now you'll notice that there are sort of other things in this transfer RNA. Like some bases are indicated and some weird things are indicated. Now, there's a C in the transfer RNA. What's that? And there's these other things. Now there's an I down here in the anticodon. What are these? These are positions in transfer RNA that get modified. So they are encoded, as normal G's, A's, U's, and C's. But certain nucleotides get modified. This is something that's very common in transfer RNAs. So how they get modified. So they get changed from the original nucleotide to something that's a bit different. And there are variable regions. In blue, these are they're in some transfer RNAs, not in others. So transfer RNAs are sort of, each of them has very distinguishing characteristics. Now this stem is important. This is the anticodon stem, and this is one of the important ones for recognition by certain enzymes to attach the amino acid, the correct amino acid, to the transfer RNA. And this anticodon down here at the bottom, this is really important. This is the part that recognizes the codon in the messenger RNA. And as I mentioned, there are lots of modified bases, and they are listed here. I can never remember these except for inosine, which is one you should know. Pseudouridine is another one that sort of sticks in my brain, but these other ones, I'm not really expecting you to know these, but you should know that inosine can be incorporated. Okay, so a big step in decoding. So the transfer RNA is important for this, but the amino acid that gets put onto the transfer RNA is really what ends up decoding the message, the messenger RNA. If the wrong amino acid gets placed onto the transfer RNA, then the transfer RNA will recognize the particular codon, but it's going to stick in the wrong amino acid. So aminoacyl tRNA synthase enzymes are responsible for attaching the correct amino acid to the correct tRNA. And this is really how the code is decoded. Right, so we have enzyme shown here. Now it's an enzyme, right? So it has a place where a specific amino acid can bind. It has it also binds ATP, so it's ATP binding pocket. So it's going to bind ATP and the amino acid, release pyrophosphate. Then it can also bind, as a place to bind, the transfer RNA. And there are different portions of the transfer RNA that are recognized by these enzymes that tell the enzyme to put to the particular amino acid onto the a very specific transfer RNA. And once this goes through this process, AMP is released, and eventually the tRNA is released that has the amino acid on it. This is called a charged transfer RNA. So there are different amino acyl tRNA synthetases that are responsible for charging the different transfer RNAs. Okay. All right, so I have a video here. The sound most likely will not play, so I will just keep quiet, and you can read the words. And I've posted this on LMS for you, so you can watch it with the sound.
Amino-acyl tRNA synthetases catalyze the formation of charged transfer RNA. To begin the process, a specific amino-acyl tRNA synthetase binds a particular amino acid and also a molecule of ATP at its active site. A covalent bond is formed between AMP and the carboxyl group of the amino acid, and pyrophosphate is released from the enzyme. A specific tRNA that has an anticodon that corresponds to the amino acid then binds to the synthetase. The RNA sequence in the anticodon region, as well as other parts of the tRNA molecule, such as the acceptor stem, are important for recognition between the tRNA and the amino-acyl tRNA synthetase. The bond is broken between the amino acid and AMP, and the AMP is released. At the same time, a covalent bond is formed between the amino acid and the 3' end of the tRNA, which is now called a charged tRNA molecule. The charged tRNA is then released from the enzyme and is now ready to be used in the process of translation on a ribosome.
Okay, so I mentioned about this wobble rule with transfer RNAs. So what is that? So we've talked about the genetic code being degenerative. So with the exception of serine, arginine, leucine, the degeneracy always occurs at the codon's third position. Now, to explain this pattern, Francis Crick proposed something called the wobble hypothesis. In the codon-anticodon recognition process, the first two positions in the codon are paired strictly according to the AU and GC rule, right? So it follows everything that we sort of know. However, in the third position, the tRNA can sort of move a little bit. So the base that's in the 5' end of the transfer RNA can wobble. And this will allow it to form different kinds of. All right, so the wobble occurs, like I mentioned, at the 3' end of the codon. This is in the mRNA, and at the 5' end of the anticodon. And it's just modified base pairing rules for hydrogen bonding at the third position of the codon. Okay, so the result of this is that one tRNA can recognize different codons. And as I mentioned, it's the anticodon can wobble so that it can recognize different codons, but the codons are going to be for the same amino acid. So here, an example of a charged leucine transfer RNA pairing with messenger RNA. So you can see there's a normal base pairing that you would expect at this position, right? This is the 5' end, right? So here's the 5' end of the transfer RNA. This is the 5' end of the transfer RNA. This is the 3' end of the messenger RNA. Right? So we get the correct base pairing. However, the same tRNA, the tRNA can change its structure slightly so that this G in the transfer RNA can also recognize uracil at the same position. Now these are, again, both leucine codons. And so this little bit of wobble that happens in the transfer RNA allows the recognition of multiple codons by the same exact transfer RNA. And so the complete set of codons can be read by much fewer number of tRNAs. And so here are the general rules. And if you're looking at the anticodon, the tRNA at the 5' end, so again, this position, G can pair with U or C. C on the transfer RNA will always pair with G. A will always pair with U. However, a U at this position can pair with either A or G. And then inosine, which is another base which is modified after the transfer RNA has been transcribed, can pair with A, U, or C. So you don't need to know the structure of inosine and the base pairing that goes on there. Just know that inosine has these options for pairing with 3 of the 4 nucleotides. All right, so let's take a look at what we sort of know so far about gene expression. So we start out here in DNA, nice double-stranded DNA molecule shown here. We have our coding strand on the top, which is going to be similar sequence to what's made in the messenger RNA, just swapping out U's for T's. Then we have the template strand shown on the bottom. So then we get down to our message right here. And we have our messenger RNA. There's some region that doesn't get translated, the 5' untranslated region. We've talked about those. You can have the same thing happen at the 3' end. These are portions that are either, in this case, on the 5' end before the start codon or after the stop codon on the 3' end. And then we have our reading frame here with codons that will be used to make the protein, make the polypeptide. These will get read by the ribosome and in conjunction with transfer RNAs, which will be able to form base pairs with each of these different codons. And this is through what we just spent a bit of time explaining, through the hydrogen bonding through the complementary between the codons and the anticodons. Now, I've talked a little bit about reading frame, and so the reading frame, right, that occurs for this protein in particular is set by the start codon, and there are other components that will help to determine that reading frame. Okay, so we finally get to the ribosome, and so ribosome is the business end of all this. It catalyzes the peptide bond formation. It's important. All right, so it consists of ribosomal proteins and ribosomal RNA, so it's both proteins and RNA. The small subunit is involved in decoding the messenger RNA. The large subunit is involved in catalyzing the peptide bond formation. And it's actually the large ribosomal RNA that provides this catalytic function. It's a ribozyme. So as we mentioned, we talked about self-splicing introns. So it's an RNA enzyme. And so there's a table from your book that sort of gives it, I'm not going to go through this in much detail, the different sort of characteristics of the ribosome. So the ribosome is composed of a small and large subunit. They have different sort of names in bacterial versus eukaryotic systems. The small subunit is the 30S subunit in bacteria, and eukaryotes is the 40S. Now what this S means is it's the sedimentation coefficient. I've talked to you a little bit about doing centrifugation in sucrose gradients, and this is just a measure of where this component sort of comes in that sucrose gradient. This is a technique that was used quite widely in early molecular biology. And so it's just listed the number of proteins. You can see that as as I've said multiple times, every time you look at eukaryotic system, it's always a bit more complex. There's more proteins, and for the small subunit, same is true for the large subunit. And they have RNAs. The RNAs are slightly larger in eukaryotes, and there's more RNAs, an additional RNA in the large subunit. And we'll talk about some of these specifically. And then when the assembled ribosome doesn't necessarily, you don't just simply add up these sedimentation coefficients to get a 30S, get an 80S assembled ribosome. Due to their interactions, it actually comes out. They will come out at a position that's sort of called 70S, and the eukaryotic is slightly bigger. Okay. So here's the structure of the ribosome, and this structure was worked out by Harry Noller. And I mentioned at the beginning of this presentation, we have our large subunit shown here, small subunit shown here. These guys here in white, right, with these bars. These are the RNA components. These purple portions are proteins, and this is also RNA over here. If you look at the small subunit, sort of this teal color, this is all, you can sort of see this is all RNA, and these are the protein parts. You can see over here, you can see alpha helices, a loop, you can see a beta sheet back here. But one of the striking features that to me, when I look at this, right, and if we're looking here at this large subunit and even small subunit, is that it's mostly RNA. So RNA makes up a large, the large portion of this enzyme. And so in 2009, there 3 people received the Nobel Prize for working out the structure of this. Okay, so in a more general sense, in a schematic nature, what do we need to know about the ribosome? So here we have our ribosome. This is our large subunit shown here. This is a small subunit shown here. Here's our messenger RNA in red. And we have our transfer RNA and the growing polypeptide chain are these little spheres coming off of the transfer RNA. So there are 3 sites in the ribosome that bind to transfer RNAs. So the A site, the aminoacyl site, accepts the incoming charge transfer RNA. The P site, or the peptidyl site, contains the transfer RNA with the growing polypeptide chain. The E site is the exit site and contains the transfer RNA that's going to be leaving the ribosome. So it's going to come in, get used, and then it leaves the ribosome. So how does the initiation start? So we'll start with talking about bacteria. So there are some initiation factors. Initiation factor 1 and 3 bind to the small subunit. All right, and we'll talk about this little pink portion here is part of the smaller ribosomal RNA, the 16S ribosomal RNA, and that's going to be important. So IF1 binds to the part that would correspond to the A site. IF3 binds to the E site. So the only thing that's available is the P site. Now, this complex here will bind to messenger RNA and sort of scan along it. And there's some things that are going to be recognized. One is that's really important, and we'll talk about this more in a few slides, is this recognition by this portion of the small ribosomal RNA. This will form base pairs with a sequence in the messenger RNA. Right? So this is shown as only 3 base pairs, but it's actually 9 nucleotides long. This is known as the Shine-Delgarno sequence or ribosome binding site. Okay. All right. So now this whole thing is, complex forms, right? And there's the initial transfer RNA is brought to the start codon with assistance of initiation factor 2, and the initiator transfer RNA is recognized as a methionine codon, but it has a different amino acid on it. It's similar to methionine. It's formal methionine. So this is the initiator transfer RNA, and this is the with a slightly different amino acid on it. Now, this is in bacteria. In eukaryotes, this is just regular old methionine. Okay, so this is the sort of we're pretty far along in initiation here. IF1 and 3 are released. IF2 hydrolyzes GTP and gets released, and then the 50S subunit associates with this. So now we have this whole 70S initiation complex where we have the complete ribosome, the messenger RNA is bound, there is a base pairing with the initiator transfer RNA in the P site with the start codon, and there's an interaction with the ribosome binding site or Shine-Delgarno site in the messenger RNA with the 16S ribosomal RNA. And so once we get here, this is the end of the initiation stage. Okay, I'm going to play another video, and again, sound won't appear on this video, but I have posted this for you. And again, you can read along. It's all written out on the bottom.
Translation initiation is a process in which mRNA, initiator tRNA, and initiation of translation in bacteria begins when the 30S, or small ribosomal subunit, binds to the mRNA near its 5' end. This process is facilitated by a Shine-Dalgarno sequence in the mRNA, which is complementary to a component of the small ribosomal subunit called 16S ribosomal RNA. Though a shorter sequence is shown here, the Shine-Dalgarno sequence is actually 9 nucleotides long. Initiation factor 3 also facilitates the binding of the mRNA to the small ribosomal subunit. Once the 30S subunit IF3 mRNA complex is formed, initiation factor 2 binds to the complex and promotes the binding of the initiator transfer RNA for N-formyl methionine to the complex. IF2 and IF3 hydrolyze GTP as an energy source to promote the association of mRNA, tRNA, and the ribosomal subunits. Translation initiation is completed when the large ribosomal subunit binds and IF2 and IF3 are released. Later, when translation is completed, IF1 is needed for the dissociation of the complex.
Okay, so that's initiation. So what's happening with sort of the elongation of translation is more amino acids get added. So here we have ribosome translating a messenger RNA. We have our transfer RNA in the P site. There are multiple amino acids have been added, and the most recently added amino acid is still attached to its transfer RNA, and it's sitting there again in the P site. So the incoming transfer RNA, so it's going to be the fourth amino acid. It comes into the A site. The growing peptide chain gets transferred to the amino acid on the transfer RNA that's in the A site. Right? This is catalyzed by the peptidyltransferase enzymatic activity of the ribosome, and this activity is performed by the 23S ribosomal RNA. So this is a ribozyme, right? That's the ribozyme action. So now this gets transferred here, and we can see it's now attached to the latest transfer RNA. So the whole peptide chain is transferred there. Now this whole thing is going to get shifted into the P site. So the ribosome translocates down 3 nucleotides, and now it sits there waiting for another transfer RNA to come into the A site. The peptide is sitting in the P site, attached to the most recent transfer RNA, and has been extended by 1 amino acid. This goes on and on until a stop codon is reached. Oh, and I should mention that we have the transfer RNA that was used previously is sitting in E site, and that will get ejected. Okay. All right, another video.
Chain elongation begins with the binding of a tRNA, which recognizes the next codon in the mRNA to the A site of the ribosome. This is catalyzed by the EFTU transcription factor and requires the hydrolysis of a GTP. Once the tRNA binds in the A site of the ribosome, the polypeptide chain is moved from the tRNA in the P site to the amino acid attached to the tRNA in the A site. Peptidyltransferase, a protein RNA complex present in the 50S ribosomal subunit, catalyzes the formation of this new peptide bond between the amino acids. The ribosome then translocates to the next codon. This process is promoted by elongation factor G and requires another GTP. This places the empty tRNA molecule in the E site of the ribosome, and moves the tRNA containing the growing polypeptide chain in the P site. The next codon in the mRNA chain is positioned in the A site. The uncharged or empty tRNA in the E site then leaves the ribosome and a cycle of chain elongation is completed. Through subsequent cycles of chain elongation, the polypeptide chain continues to elongate one amino acid at a time.
Okay, so translation has occurred. It started, it's been going for a while. And what happens when it wants to stop? Okay, so here we have our ribosome. It's translating and then all of a sudden it comes to a stop codon. Now there's no transfer RNA to recognize that stop codon that's located in the A site. So we have the last transfer RNA that was used with the now finished polypeptide chain connected to it sitting in the P site, and the E site should be empty. So there's a release factor. We'll bind to this, bind in the A site. So release factor 1 or 2, these recognize stop codons. So there are proteins that bind into the ribosome when the ribosome is stalled, so sitting here at a stop codon. The polypeptide gets cleaved off of the transfer RNA and released. The transfer RNA also gets released. Eventually, the ribosomal subunits, the messenger RNA, and the release factor also get released. And all these components are now dissociated from one another and available to go. Okay, so I mentioned that we're going to, some things about translation initiation. And so I'm going to go and take this time now to talk about the differences in translation initiation between bacterial or prokaryotic systems and eukaryotic systems. So the question is, how does ribosome pick the appropriate start codon, the appropriate AUG, from all the other AUGs that are sitting in a messenger RNA that are just supposed to code methionine? So in bacteria, keep in mind that we have a messenger RNA where there can be multiple genes sitting in that mRNA transcript. And so the ribosome is going to have to know to start translating, at this beginning of this blue portion, and also at the beginning of this blue portion that's more downstream to make both of the proteins that are on this messenger RNA. So there are start codons in both of these, at the beginning of both of these coding sequences, and there's a Shine-Dalgarno sequence which is upstream from those. Right? So to remind you, this is a sequence that exists in the messenger RNA. The Shine-Dalgarno sequence is in the messenger RNA, and it will form a base pair with a portion of the 16S ribosomal RNA in the small 30S subunit. So here we have our start codon, then we have just random sequence here, and then we'll see a Shine-Dalgarno sequence that's usually very purine rich. This AAG. And when you actually look for Shine-Dalgarnos in messenger RNAs, they look, they can vary quite considerably from the optimal sequence. So what does this do? This sequence here in the messenger RNA is bound by a complementary sequence in the ribosomal RNA. So it's these two pieces, the start codon and this Shine-Dalgarno, that designate where a start codon is going to occur in a bacterial messenger RNA. What about eukaryotes? So they have a couple different ways to do this. Right? So how do they pick the correct start codon? So there's a cap-dependent version. So remember, cap is the 5' 7-methyl guanine residue that's attached to the 5' end of the messenger RNA. Well, this can be recognized. So we have the small subunit and many subunit and initiation factors bind to the cap and scan for the first AUG that it comes across. Now, it's not that simple. There's also more, there can also be AUGs with some more information around them. So there's a sequence called the Kozak sequence that surrounds the appropriate start codon. And so you have AUG here in green. The Kozak sequence would be these additional nucleotides, ACC, AUG, and G that surround this. This helps to distinguish what the right AUG triplet is for starting translation. Now, there is another way that eukaryotes can initiate translation, and that's called the cap-independent method. So it doesn't, here the ribosome doesn't recognize the 5' cap. Instead, finds a position inside the messenger RNA. So this is what's called an internal ribosome entry site, or IRES. Right? So it just means that there's some portion in the messenger RNA that gets recognized. Now, this site is usually a very structured portion of the RNA. It has stem loops, and this structure can be bound by the small subunit and the initiation factors. So for cap-dependent, which we've talked about, it looks something like this. Here's our AUG. Here's the cap. This gets recognized by a bunch of other factors, which you don't really need to know all of these. We have talked about poly binding protein, and so this is sort of links the 5' end to the 3' end to the poly A tail. Right? So this brings the ribosome in, and it searches for this Kozak sequence, and it will start. Now for cap independent, there's a structure here in the RNA. So the 5' end is located somewhere over here. There's no scanning looking for the correct start codon. Instead, there are factors that help bring the ribosome to this position, to this structured portion of the RNA. Now, something to note is that the 3' end here with the poly A tail is also connected to this, right? So we still have poly A binding protein associated with this. But now there's a start codon that's somewhere in the middle of the messenger RNA, and that gets that's where translation initiates. So there's no scanning, and there are fewer factors involved. Now, one of the neatest things for me about translation is that once an mRNA is available, a ribosome can get onto the start codon and start translating. As soon as it clears that initiation sequence, another ribosome can get on there. So you can have multiple ribosomes on the same message, all in different states of translation. So here's one that's just starting. There's no protein here. 3 amino acids have been added. This one is further down. More amino acids have been added. You go all the way down and around over here. Eventually it gets to the stop codon, and the whole the whole polypeptide has been synthesized. All the amino acids in there, and you can actually see this with electron microscopy. So I've always found, so these are called polyribosomes, and these are what sort of the active version of translation of the ribosomes are exist as. So you have individual ribosomes. They're usually not translating anything. It's usually multiple ribosomes associated with a messenger RNA. Okay, so let's take a quick summary about gene expression in eukaryotes and prokaryotes. So eukaryotes, we saw that we have our DNA in a nucleus. It has exons and introns that need to get processed. So we get transcription into messenger RNA, and we have those exon and introns in the message. They get removed. 5' cap gets added. Polyadenylation, so you get the poly A tail, and this whole thing gets exported out of the nucleus, and then this will get translated. Now in bacteria, we start with DNA. Usually there are no introns. This gets transcribed into messenger RNA and gets translated into protein. Now what's interesting here is that this all happens in the same compartment. There's only one compartment in a bacterial cell. In a eukaryotic cell, transcription and processing take place in the nucleus, and translation takes place in the cytoplasm. So this can, when we talk about regulation, this can lead to different forms of gene regulation, because as soon as the messenger RNA is made, even before it's finished, ribosomes can jump onto it and start to translate it. So that's another interesting feature. You can have polyribosome messenger RNAs that aren't even finished being transcribed. Okay, so what do we know about ribosome and disease. So there are a few diseases associated with the ribosomes, and this is not meant for you to know all these, and it's probably very difficult to read on this. But I have a reference here if you want, if you're interested. And so translation-related diseases are generally very rare because it's such an important process that usually defects in translation are going to end up being lethal. But they can affect initiation, elongation, mitochondrial translation, and they can also affect the amino acyl tRNA synthetases. And they can have specific effects on translation of specific messenger RNAs. So here we have our, just a sort of standard messenger RNA in eukaryotes. We have our 5' and 3' untranslated regions. This can be bound by proteins. It can form stem loops or more complex structures, and those stem loops are often involved in regulation. So you can get different RNA structures forming throughout this messenger RNA. So there's a certain syndrome, hereditary hyperferritinemia cataract syndrome, where you get excess iron storage for the protein ferritin. Patients develop cataracts even as infants. And it has to do with the change in the 5' untranslated region of the ferritin messenger RNA that prevents inhibition of translation. So normally, here's our messenger RNA. Here's the 5' untranslated region. We have a ribosome and initiation factors binding to the cap and scanning, and here's a start codon. There's a protein that sits there and prevents the ribosome from getting to the start codon. All right, and this is regulated, and so when translation needs to occur of this gene, this protein gets removed. However, there are mutations that occur in the messenger RNA in this sort of region. So there's a stem loop that gets bound by this protein that changes that stem loop, prevents binding of this inhibitory protein, and you get translation of this of the ferritin mRNA more than would normally be ideal, and that's what causes this disease. And then there are some melanomas, skin cancers, where mutations in the 5' untranslated region create false start codons. This impairs the ribosome scanning for the correct start codon. It finds false start codons. And this reduces translation. And when this happens in particular transcripts, the one for a P16 tumor suppressor, it reduces the translation of that. That protein is usually involved in suppressing tumors. And now if that's not made as much, tumors have a greater chance to proliferate. And so we have, normally we have the ribosome binding at the 5' cap and scanning along until it finds the right start codon. Well, now all of a sudden there's a mutation that creates a different start codon, and the ribosome runs to that. It starts to translate that. It may not do it 100% of the time. It may make it past this, and this might actually be a better start codon. It may have a better Kozak sequence surrounding it. But any sort of AUG can be a substrate for translation initiation, after the 5' cap. So it's going to reduce the amount of translation of this message. Okay, so that's going to end our talk on translation, and we will see you next time.